Good morning, everyone. First, I would like to thank the Microbiology Consortium of the Philippine Society for Microbiology Incorporated for this opportunity to share with all of you what I know about the domain Archaea. The archaeal cells are perhaps the most ancient cells that exist on Earth as depicted by this comic strip. Under the microscope, they do look like the bacteria, as they are unicellular and can appear as coccus, rods, and even form clusters, as we see in some bacterial cells. However, as I go on with my talk, you will realize that the archaea are quite distinct from the bacteria. Archaea is a huge topic that I cannot cover in this single talk. In this talk, I will focus on the following. I will briefly describe the history of the discovery of the domain Archaea, show you the expanding tree of the domain, discuss some of their importance, compare them against bacteria, discuss some of their metabolic strategies, and identify some research opportunities. Before the 1977, the Archaea were unknown to us, or more accurately, they have been in existence but were hidden before us. This is a copy of the program of a symposium that I attended in 2007 as a PhD student at the University of Illinois to then celebrate the then 30th anniversary of the discovery of the domain Archaea. I got myself a souvenir shirt that I will treasure forever. The first member of the Archaea was described as early as in 1880s, but of course it was not referred to as Archaea then. Methanogens were isolated in 1936. Even before 1970, Carl Woese was already studying evolution. In 1977, with George Fox, Woese published a paper revealing the presence of the third domain of life, on top of domains bacteria and eukarya. In 1990, the domain Archaea was formally proposed. The first complete genome of an archaeon was released in 1996, a methanogen named Methanococcus yanashii. In 2008, the upper limit for life was seen at 122 degrees Celsius in a methanogen. Thereafter, several more genomes of Archaea have been sequenced and deposited in public databases. In the late 1960s, Carl Woese was investigating the evolution of life using the small subunit of the ribosomal RNA. His protocol involved digestion and 2D electrophoresis of the digested fragments. The generated fingerprint or sequences were then compared and analyzed using a computer program developed by George Fox. This is one of the fingerprints that they generated. Archaea were included in the analysis of Woese because Dr. Ralph Wolf of U of I was then working on the biochemistry of the methanogens. He observed some unusual features in the methanogens that were not found in bacteria and believed that they could be distinct microorganisms. He supplied these cultures to Woese during his study. Now Woese, or Wolf was the mentor of Dr. Metcalf, who worked on the development of genetic techniques for the methanogens. Dr. Metcalf was my advisor when I pursued my PhD at the University of Illinois. So that is my very short and very weak connection to the discoverer of the domain Archaea. Please forgive my indulgence. The breaking paper of Woos and Fox was published in 1977 in PNAS. Remarkably, the paper contains a single table showing the association coefficients or similarities between the rRNA sequences of only 13 organisms. The table shows three distinct clusters of similar scores, one for the known eukaryotes, another for the known bacteria, and another distinct cluster for the methanogens. Based on these findings, the authors propose three lines of descent for all living organisms, namely bacteria, Archaea and the eukaryotes. These findings made headlines in the New York Times with the younger Woos on the cover. This is a pedestal at the University of Illinois commemorating the archaea that Woos discovered. The tree shows that the archaea, although prokaryotic, are more closely related to the eukarya than to the bacteria. However, phylogenomics says that there could be not three, but just two domains of life, where the eukarya evolved from the archaea. However, let us debate on this some other day. The fact remains that archaea is a distinct domain, whatever tree of life you prefer. The earlier phylogenetic tree of archaea shows five phyla, namely the eukaryota, which contains the methanogens, the nanoarchaeota, which contains some parasitic archaea, the core archaeota, the cren archaeota, which includes known thermophiles, and the thome archaeota that includes some mesophiles and nitrifiers. However, the advent of genomics has led to the discovery of several new taxa, expanding the tree of archaea. As of 2017, several new phyla, classes, orders, and family have been discovered. New superphyla have also been discovered, including Asgard, Tak, and Dipan. Many members of the domain Archaea are extremophiles, but not all Archaea are extremophiles, and vice versa. Hyperthermophiles, psychrophiles, pisophiles, halophiles, acidophiles, and radiation-resistant species have all been found in the domain Archaea. Several members of the domain Archaea play important roles in various biogeochemical pathways, such as the sulfur, carbon, and nitrogen cycles. Some are major players. For example, the methanogens are the major players in the formation of methane from CO2. Several more archaea play an important role in the oxidation of hydrogen sulfide to sulfate, as well as the oxidation of ammonia to nitrate. Some groups of archaea have been associated with various parts of the human body, such as the nose, lungs, skin, and the GI tract. Examples include the methanogens, which are abundant in the nose and gut of the human body. Members of the phylum Thome archaeota are abundant on the skin, while the Woos archaeota are abundant in the lungs. However, There is no conclusive evidence to prove that pathogenic archaea exist. Under the microscope, some archaeal cells resemble the bacterial cells as they can be rod and coccus, but unusual shapes like branching cells and square cells have also been found among the domain Archaea. Some unique cellular structures have also been found among Archaea. One of these is the cannula. The cannula are hollow tubes that connect the cells of the archaeon pyridixium. The cannula is embedded in the periplasm of the cell, but it is unclear whether it is used for the exchange of nutrients and genetic materials between cells or provide a means of attachment and anchoring. The hami are a new class of filamentous surface appendages from an uncultivated euryarchaeal coccus, living with bacteria in cold sulfurous marsh water. They attach on bacterial filaments and divided into two regions, namely the central part with a barb-like structure and the distal part or the hook region with a tripartite end. The flagellum of the bacteria and of the archaea, which is called archaeolum, are quite distinct from each other. The filament of archaeolum is much thinner, and for example, in Halobacterium species, they swim at only 2 to 3 micrometers per second against E. coli cells that can swim as fast as 25 to 35 micrometers per second. The archaeolum is used for motility, adhesion, biofilm formation, and other symbiotic and environmental activities. The proteins making up the archaeolums are found to have widespread end-linked glycosylation. The interior of the archaeolum is similar to type 4 pilus and it lacks hollow interior. Therefore, the archaeolum subunits are predicted to assemble at the base. The flagellines, which are the subunits making up the archaeolum, exhibit no similarity to the bacterial flagellines. They also have signal peptides similar to the type 4 pilus. The cell walls of bacteria and archaea are also distinct from each other. While two major types of cell walls are found in bacteria, the gram-positive cell wall and the gram-negative cell wall, there are various types of cell walls found in the domain Archaea. For example, in Sulfolobales, S-layer functions as the cell wall. In others, it's an outer membrane. In Methanosphera, a pseudomurine functions as the cell wall. In Methanothermus, there are two layers, a layer of pseudomurine and S-layer. In Methanospirillum, there is an S-layer and another sheath. In Methanosarcina, there is a layer of methanochondroitin and another layer of S-layer. All bacterial peptidoglycan contain N-acetylmuramic acid linked to N-acetylglucosamine by a beta-1,4 glycosidic band, which is sensitive to lysozyme. In contrast, the archaeal pseudomurine contains N-acetyltalosamine acid linked to N-acetylglucosamine by a beta-1,3 glycosidic band, which is not sensitive to lysozyme. Now, I have some questions to the audience. Can we subject archaeal cells to gram staining? If no, why not? If yes, can we now use the gram staining result as a taxonomic marker in Archaea? So this is something that we need to think about. The bacteria and Archaea also differ in their cell membranes. The phospholipids of Archaea are made up of branched isoprenoid chains, ether linked to glycerol. The phospholipids of bacterial cell membrane, on the other hand, are made up of fatty acids, ester linked to glycerol. In addition, bacteria and eukaryotes form lipid bilayer, while some archaea form a lipid monolayer. Transcription in archaea is similar to the eukaryotes. The RNA polymerase A of archaea requires basal factors to recognize the promoter region. The basal factors include the tata-binding protein or TBP and the transcription factor B or TFB, which both have eukaryotic homologs. In contrast, the bacterial RNA polymerase requires a sigma factor to recognize the promoter region to initiate transcription. The RNA polymerase of archaea resembles the RNA polymerase II of the eukaryotes. While the bacterial RNA polymerase contains 5 subunits, the RNA polymerase of the archaea contains much more number of subunits, 13 of them, 11 of which have homologs of the eukaryotic RNA polymerase II. The core components and complexity of translation in archaea are eukaryotic in nature. The archaea also exhibit elaborate translation initiation process requiring at least 5 initiation factors, which again have eukaryotic homologs. Now going to some information on microbial genetics in archaea. Various DNA transfer mechanisms have been observed in archaea, including those found in bacteria such as transformation, transduction, and conjugation. In addition to this, in the order Thermococcales, some cells release vesicles. They release vesicles that contain either chromosomal or plasmid DNA. These vesicles can then fuse with recipient cells, transferring the DNA that they carry. In cell fusion, cells establish physical contact by creation of cytoplasmic bridges. This creation of contact forms heterodiploid cells. These heterodiploid cells contain 2 different chromosome types from the parents and all the plasmids combined. This heterodiploid cell will lead to either segregation of the chromosomes and plasmid into the original state or the parents, or recombination events between the chromosomes that would create a hybrid of the parental strains. In addition, the plasmids can be exchanged or they can also be divided unequally. In Cran archaeota, when cells aggregate, chromosomal DNA can be transferred by crossing the recipient membrane through a channel. This will then be followed by incorporation of the chromosomal DNA into the genome of the recipient cell through homologous recombination. In the laboratory, several natural and artificial delivery systems have also been developed for the archaea. These are the use of polyethylene glycol, electroporation method, the use of liposomes, chemical transformation using calcium chloride and heat shock, and the natural DNA transfer methods like conjugation and transduction. Let me focus on the liposomes. Liposomes are spherical vesicles made up of phospholipids. They form a lipid bilayer. Liposomes can enclose DNA and they can fuse with the cell membrane of the recipient cell, releasing the DNA that they carry to the cytoplasm. We now proceed to the carbohydrate metabolism in archaea. Shout out to all my former students in MCB 120, Microbial Physiology. Some bacteria utilize the Embden-Meyerhof-Barnes pathway, where glucose is phosphorylated to glucose 6-phosphate. It is then isomerized to fructose 6-phosphate and phosphorylated to form fructose 1,6-bisphosphate, a C6 carbon. This C6 carbon is then split into two C3 carbon compounds, namely the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, or more popularly known as pGALD. In the lower shunt of the pathway, pGALD or GAP is then phosphorylated to 1,3-bisphosphoglycerate, which is a high-energy molecule that serves as a phosphoryl donor in the synthesis of ATP via the substrate level phosphorylation. 3-PG is then converted to 2-phosphoglycerate, which is then oxidized to phosphoenol pyruvate. Phosphoenol pyruvate is a high-energy molecule that donates its phosphate group to ADP to form ATP via again the substrate level phosphorylation. Pyruvate is another product of the reaction. In archaea, although catalyzing the same conversions, the enzymes in the EMP pathway versions differ remarkably. The pathway modifications occur mainly in the upper part of the archaeal pathways involving an ADP or PPI or ATP-dependent kinases, distinct from the classical enzymes in bacteria and eukarya. The most striking difference in archaeal pathways, particularly in hyperthermophiles, is the direct and irreversible oxidation of glyceraldehyde 3-phosphate to 3-phosphoglycerate. Bypassing these reactions in the classical EMP pathway to produce 3-phosphoglycerate. This is catalyzed by an enzyme which is NADP plus dependent glyceraldehyde 3-phosphate dehydrogenase, GAPN, or mainly found in anaerobes by a ferredoxin-dependent glyceraldehyde 3-phosphate oxidoreductase, or GAPOR. Both enzymes omit the formation of 1,3-bisphosphoglycerate and the production of ATP via substrate level phosphorylation, with considerable consequences for energetics. Now we proceed to the Ettner-Duderoff pathway. In the classical Ettner-Duderoff pathway found in bacteria, The glucose 6-phosphate is oxidized to 6-phosphogluconate, which is then dehydrated to 2-keto-3-deoxy 6-phosphogluconate or KDPG. KDPG is then split into pyruvate and glyceraldehyde 3-phosphate. Now GAP is further metabolized to produce pyruvate using the same reactions as in the classical EMP pathway. Archaea utilized the modified Etnar-Duderoff pathways, the non-phosphorylated ED pathway, and the semi-phosphorylated ED pathway. In these modified versions, the following reactions do not occur. In the semi-phosphorylated ED pathway, glucose is directly oxidized to gluconate. Gluconate is then converted to 2-keto-3-deoxygluconate or KDG, which is then phosphorylated to KDPG, which is then cleaved to the glyceraldehyde 3-phosphate and pyruvate. Glyceraldehyde 3-phosphate then enters the lower common shunt of the EMP pathway. Thus, the semi-phosphorylated ED pathway has the same ATP yield as the classical ED pathway in bacteria. In the non-phosphorylated ED variant, KDG is not phosphorylated but is directly cleaved into pyruvate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is then phosphorylated into 2-phosphoglycerate and further converted to a second molecule of pyruvate. Due to bypassing the phosphorylation at the level of KDG and also subsequent GAP oxidation, and since GAP oxidation to glycerate is not coupled to ATP formation, the net ATP yield of the non-phosphorylated ED pathway branch is 0. Let us now focus on the methanogens, as these were the organisms I worked on for my PhD. The methanogenic archaea are biological agents of methanogenesis, the production of methane, which is a potent greenhouse gas and a potential renewable energy source for heat, electricity, and transportation. The methanogenic archaea produce approximately 400 million metric tons of methane annually. So this is a lot of energy. They are all obligate anaerobes. The Methanosarsinae species in particular are metabolically and physiologically the most versatile methanogens. They can produce methane from no less than 9 substrates, including hydrogen gas and carbon dioxide, acetate, and several C1 compounds such as the methylamines, methyl sulfides, and methanol. The Methanosarsinae species utilize 4 overlapping methanogenic pathways to produce methane. The CO2 reduction pathway involves the reduction of carbon dioxide to methane using hydrogen gas as the source of electron. The methyl reduction pathway also uses hydrogen gas as an electron donor but reduces methanol to methane after the transfer of the methyl group to the coenzyme M. In the methylotrophic pathway, the methanol, for example, is disproportionated into carbon dioxide and methane. The oxidation to carbon dioxide produces reductants that are then used to reduce one molecule of the methyl group to produce one molecule of methane. In the acetoclastic pathway, acetate is first activated into acetyl-CoA, and then acetyl-CoA is dismutated into carbonyl moiety and the methyl moiety. The carbonyl group is then oxidized to carbon dioxide, whereas the methyl group is then reduced to methane. In Archaea, that derive energy from hydrogen such as the methanogens. Electrons originating from hydrogen gas reduce the F420 and then the methanophenazine. The latter, through the cytochrome B type, reduces the heterodisulfide reductase with the extrusion of protons to the outside of the membrane. This extrusion of protons creates a membrane potential that can be used by ATPase to synthesize ATP. In bacteria, quinones and other cytochromes are used to shuttle the electrons from hydrogen gas, also producing a proton motive force used to make ATP by ATPase. Let us take a look at the ATPase of the Archaea. The A1, A0 ATP synthase is present in Archaea. The V1, VO ATPase is found in the eukarya. The F1, F0 ATP synthase is found in bacteria, mitochondria, and chloroplasts. The common feature of these ATPases is a structure made of two rotary motors, the 1 and the 0 motors, connected by a central stalk and 1 to 3 peripheral stalks. A major functional difference is that the predominant physiological function of A1, A0 and F1, F0 is to synthesize ATP at the expense of an electrochemical ion gradient, whereas the V1, VO ATPase drives ion transport at the expense of ATP hydrolysis. To recap what we have discussed so far, we talk about the history of the discovery of the domain Archaea, the expanding tree of the domain, their importance, comparison against bacteria, their metabolic strategies, such as glucose degradation pathways, methanogenesis, electron transport chain, and ATPase. Archaea offers a wide range of research opportunities, such as revealing the diversity of the domain, particularly those in extreme environments, using molecular approaches such as metagenomics, single-cell genomes, etc., and then determine the forces that drive such diversity. Another is to unravel novel metabolic pathways, discover ecological significance, and understand the molecular basis of survival of the extremophilic Archaea in harsh environments such as the molecular basis at the level of DNA, RNA, and proteins. Another possible research opportunity is to demonstrate the underlying process of gene flow. And then for application purposes, explore industrial and biotechnical applications of the archaea. For example, novel enzymes and other biomolecules of the thermophilic, acidophilic, halophilic archaea. And then another research is to investigate whether extraterrestrial life exists. This is the field we call astrobiology. You can then present your research in a conference exclusive for domain archaea. One of these is the Archaea, Ecology, Metabolism, and Molecular Biology, such as this one that I was honored to participate in. The conference gave me the opportunity to meet and talk with several Archaea experts. Many of the works that I cited in this talk are from the participants of this conference, such as Dr. Cybers, Dr. Jarrell, Dr. Caviccioli, Dr. Whitman, Dr. Allers, of course, my supervisor, Dr. Metcalf, and Dr. Albers. The conference is held only every 2 years. This year, it will be held at West Dover, Vermont, USA. With this, I would like to end my talk, and I am now ready to accept questions from the audience. Thank you very much.